that better? Okay. So last week we talked about psychopharmacology. That was a lot of fun. We talked about a few other things. Today I would like to briefly talk about methods. Right? I'm not expecting you to memorize these methods. Okay, I think that's probably a little bit beyond what you'll need to do. I'm not expecting you to fully understand what's going on with any of these particular methods, but I think it is a nice idea to have some sort of background, right? So later when we talk about these different studies, we talk about the different research programs and experiments, you have a little bit of an idea, you know, what they're talking about, what they did, how difficult this was. Who remembers when we talked about Pierre Florenz? I know was that anybody's favorite, you know, French neuroscientist? No? Okay. Yeah. You probably only know one, right? So he's got to be your favorite. Well, I guess you've got Broca, so you can kind of pick there, right? Experimental ablation. We're going to talk a little bit about recording and stimulating neural activity. We'll talk about neurochemical methods. And then we'll actually have a small conversation about genetic methods as well. There's some cool stuff there. All right, brain lesions, right? How many of you want to produce brain lesions in people? Right, yeah, right? I mean, this is a lot of fun. Okay. There are a few ways you can do this. You can use the old you know, spork method of Pierre Florenz. You can just scoop out parts of someone's brain, right? That's always exciting. So actually, feel free to do that, right? I've got cards I can give you. I'll sign the back. You'll be authorized. It's perfectly fine. But there are other ways that we, in the last 200 years, we found better ways of removing parts of your brain, right? Or the parts of brain of experimental animals, because we decided, well, using spoons is it's a little inaccurate. Right, so maybe we should try something better. We use radio frequency. That's kind of fun, right? Just blast somebody with a radio frequency and kill part of their brain. So that's fun. Oh, well, what about excitotoxic? That's cool too, right? This is when you squirt in some sort of chemical. What's an excitatory neurotransmitter that we could squirt in there that might be exciting, right? That would like overexcite a cell. Something like glutamate, for example, or glutamate agonists, right? To use a glutamate agonist that would increase brain activity to the point that those cells die. I do want to kind of give you a word of warning here. You can't, like, you know, think or study so hard that you start to kill your brain cells. I know some of you might try to use that as an excuse, right? You were afraid of creating excitotoxic lesions, and that's why you didn't put the full effort into this class. It's not going to happen, right? You really have to use some special chemicals to make that happen. So yeah, keep taking notes on that. That's important. Okay. And then sometimes you should do a sham lesion, right? And I think this is an important part about experimental studies. You always have to have a control group. Okay. How many of you have taken like the experimental psych class? Okay, like one of you wants to admit that. That's fine. Okay, 3. Thanks for. Anytime you do an experiment, you need a control group, right? And if you're going to crack open somebody's head and you're going to squirt in a chemical that's going to kill brain cells, that seems rather invasive, right? So maybe that's going to cause you some problems. Don't have to answer this. Has anybody ever had a hole in their skull? Damage to their skull? Trust me, it can cause you problems, right? It really can. So we might want to have a control group. We'll do a sham lesion. Typically this is going to be saline. Okay, just salt water. Anybody used saline before? Anybody that has contact lenses? There you go, right? It's good to drink. It's very tasty. It's not very tasty. You can use it as a like a gargle or a rinse, right? Not a lot you really need to take away from this particular image. This, by the way, is your hippocampus. Okay. Well, it's the hippocampus of a rat. It's not really your hippocampus. And if you were to look very, very closely, you could see there's like a little hole right there that was created with glutamate, actually, that excitatory amino acid. All right. So your brain is a three-dimensional structure, right? And it's not the same all the way through, right? If you think about, how many of you have ever seen a loaf of bread? Yeah, like regular white bread, right? Not talking like the fancy bread that's got stuff in it, but a regular loaf of white bread, it's like all the same all the way through. No matter how you slice that, you're getting different shaped slices, but the inside's all the same, right? It's just bread all the way through. Your brain's not really like that. While it is sort of brain all the way through, we hope, right, with a couple exceptions for your ventricles, the brain regions, they're all different as you go through, right? It's different brain nuclei. And so we want to make sure, if we're wanting to know, what do we want to know about fear responses in a particular situation, we want to make sure we're actually hitting the fear nucleus, right? We want to make sure we're actually hitting the amygdala, and we're not just like randomly like jabbing into a brain, right, while somebody's like wiggling around. So we do something called stereotaxic surgery. We use this sort of three-dimensional coordinate system. Okay, you can create maps. This is the map of a rat brain, right? This is kind of a coronal slice. So imagine the rat nose like coming out like from the screen, right, trying to grab you. No, it's really going to give you nightmares tonight. And you would move around and then lower into the target. Not a big deal, right? Here's what a rat looks like if it was in one of these devices, right? So you have these sort of three-dimensional movement. Okay, so you can locate any spot in the rat brain. Some of you might think, well, why would I want to do this to a rat? There are a lot of good reasons, right? So we'll talk about those later. You might also want to do this to a person. Okay, why would you want to do this to a person? Well, perhaps they have something in there that needs removed, right? So if you think about some kind of removal of brain tissue or a tumor, you might want to try to figure out what's going on. So I think that's sort of a interesting photograph for you. Has anybody ever seen a brain that's not been, or you got one of these at home? Well, they're popular, you know. They're fun devices. Yeah, it's a good Halloween costume, right? He goes, brain surgery man. I don't know if that's a thing. Please don't tell me your Halloween costumes. I don't care. Kyle, I've had one set of trick-or-treaters my entire life of living anywhere. This was when I had an apartment when I was in college. I don't know why they... I mean, like, if I was going to go somewhere, I think I might, right? Because college kids would give away interesting things, right? So, like, as an adult, I think that's maybe where I would go trick-or-treating. I gave away plastic straws. Not like ones from McDonald's, but so my wife and I, we bought these, like, like spider rings and like straws with pumpkins and things. I don't know, right? They were very popular. We had kids coming back like, hey, I heard you had straws. It's like, I don't know what kids are doing with straws these days, but here you go. Vampire teeth, we gave some of those away. Trying to do something, we don't want kids eating candy, right? We just want them choking on vampire teeth. I mean, if you make them sick, they might come back, right? Choke. It is. It's a completely different story. It really is. So how many of you have ever cracked open the head of an animal? No, nobody's done that, right? Anybody ever seen a brain that's like, yeah, was this in or out of something? Well, tell me the context. Are you allowed to tell me the context?
It was a cow.
It was a cow? Okay.
And I was doing it for something in the science department.
Did it smell bad? Yeah, so they had already done something to it before you got it, right? It wasn't just like the cow wasn't like walking around and then you just like did the convertible job on its head. No, that's not what you did. It had probably already been through a process called fixation, right? And so basically there's like formalin or some formaldehyde derivative they will put through the body so that the brain becomes a bit more firm, so you can actually manipulate that, right? If you get your hands on a brain before this has happened, it's really squishy, right? You'll just like grab it and it's like squish between your fingers, right? Isn't that fascinating? You have to be very careful with your brains. That's why all of you came equipped with this awesome safety device. called the skull, right? Why in the world would we put such thick bone around something if it wasn't soft and squishy and easily damaged, right? We wouldn't. We wouldn't waste our time on that, right? If something like your liver was that delicate and that important to your survival, what would you do? You'd have a big, you know, bone plate for your abdomen, right? Seriously. Makes sense. So there you go. And then you have to cut it with one of these things, right? Because you have to slice it to take a look at it. So when we look at these brain slices, how many of you have been to the deli? Yeah, it's kind of like one of those things, right? But it's a really, really sharp blade. In fact, they might even, if you want to cut something really, really thin, I'm talking like just a few atoms, right? Like we're not even measuring like microns or micrometers anymore. You might actually use a diamond knife, right? So that sounds exciting. Then you have to get one of those things sharpened. And if you're good, you end up with something that looks at least as good as this. Okay? This has been stained with something called Creswell violet stain. Don't worry about what that is. Basically, it turns all the cell bodies purple. So that's exciting. And you can go, hey, there's a brain cell body. Isn't that exciting? And we learned a few weeks ago those are called, it's called soma, right? Anybody used the microscope before? Right? Those are fun, right? Anybody still have a microscope? So many fun things you can do with that, right? Like, I mean, like look at things that are small. I think that's really the list of what you can do. They make some other microscopes that we might talk about. One of those is an electron microscope. This you actually shoot electrons at something. That sounds really exciting, right? So you shoot electrons through brain tissue. You got to have a little piece of that. And then there's also like, who loves lasers? Nobody's a big laser fan. I mean, go to laser light shows. You make your cat run into a wall with a laser, right? You guys do all the, I know. So there's laser microscopes as well. You can see things that are fluorescent. This is kind of cool. This is from an electron microscope, right? And this is a terminal button, like a terminal. And these little fuzzy things here, those are arrows. They're not really in your brain, right? But these two little fuzzy patches, those are actually synapses, right? And so at the synapse, there's a ton of proteins. One of these proteins that we've talked about before, Ellis, is the receptors, right? So you've got a bunch of receptors there. We call that a postsynaptic density. That's a fancy term for saying, hey, if you look through an electron microscope, there's a fuzzy patch right there. And that's where this terminal is contacting, or this terminal, sorry, is contacting onto this axon. This is an axon here. And this one up here is the terminal. These guys are mitochondria. Don't worry too much about those. It's kind of fun, right? I always like to point out, this really irritates me because this is like from your book publisher, right? This image. But there is an air bubble right there. See that circle? I mean, if you're going to take the time to write, don't show something with air bubbles. It's just not good. This is from a scanning electron microscope, and then they've added some funky colors to it just to make make things pop out. Not a big deal. Again, we're dealing with a cell body there. We've got dendrites and axons coming out. Okay. How many of you love food coloring? Anybody ever use food coloring? Right? You can make things that are like food, different colors with food coloring. That's what it does. There's stuff that we would use. I call it scientific food coloring. Basically, if you have a brain region, you put some of it in there, you wait a little bit, and you see where it goes. And you can figure out which brain regions are connected, right? Because, again, it's all just like a big wad of juicy fruit. It's kind of hard to, like, trace, like, individual neurons any other way, right? So you have to get a little more technical. Stuff goes left and right or front and back. Don't worry about it too much. Don't worry about that. Who loves rabies? Yeah, right? Rabies is pretty awesome. Anybody had the rabies vaccine? No? Nobody's. People are thinking about that. It's not fun. I had the rabies vaccine, Kaylin, a few years ago. As a preventative, you never know, right? I thought it was going to work with ferrets once, and because ferrets are known to carry rabies, and so I had to get the rabies vaccine. It was like 3 times I had to get a shot. It was actually pretty painful. My whole side hurt. And then so at some random time in the future, I might actually develop rabies from this. So that's a warning to all of you. Like if I really just start foaming at the mouth and acting strange, you know what the problem is, right? So rabies is cool. Why is rabies cool? Let's get this to stop. What is a virus really great at doing? A virus is, you're going to say, well, it makes me sick. Well, yeah, it does that, right? But a virus is really great at getting into your cells and then like making that cell, it tricks it into making its own DNA, right? And then it can move on to other cells. So that's pretty cool, right? And so if you knock off the part of the virus that's actually like the part that makes you sick, right? You can use this in the brain because you can hop into like one brain cell and then you can follow that rabies virus, or they'll also use like herpes, right? So if you don't love rabies, I know you love herpes, you can follow the movement of that virus molecule. See, Kyle, these are the great things that you can pick up and use in classes when you teach, right? Yeah. So you can follow that rabies virus or that virus molecule as it moves. That's kind of cool, right? Don't worry about this. Don't worry about this. We're just going to skip through this. So some of you might not really be that interested in like cracking open heads and doing things, right? And there aren't a lot of people signing up for those kinds of studies, right? Well, nobody's like, hey, squirt some rabies in there. Let's see what happens. I could just imagine like advertising for that study, right? Who wants rabies squirted in their brain? That's why I don't get things through the IRB that easily. So maybe there's some things that you want to develop, like, well, I'd rather not cut someone's head open. And maybe they won't let me cut their head open, but they'll let me slide them into a tube, right, and shoot magnets at them or something, right? And so we'll do that. And so that's what folks have done with CT scans. You don't have to answer this. How many of you have had a CT scan? I'd say a number of you have, or you know someone who's had a CT scan, right? Not necessarily on your brain, but they do them other places. What about an MRI? MRIs are fairly common, right? They use some magnets to get an image of the inside of your body. And there's another one called diffusion tensor imaging, DTI. This one's kind of cool. It looks at water molecules. This is really great at tracing like fiber bundles, right? So bundles of axons. It looks at the orientation of the water molecules in these myelinated axons, and you can map that out, which is kind of cool. You remember that fun multicolored spaghetti picture I showed you last week or the week before about connections between through the corpus callosum? Nobody remembers. There's video evidence of that I showed you that, so you can go back and look at that, right, Kyle? Pull up that YouTube video, make sure I did that. Here's somebody going into like a CT machine. Here are the type of brain scans you would get out of that, right, which are kind of interesting. You can see some cool structures. Hey, what are these 2 circles right here? Those are the eyes, just to give you an idea. Here are some ventricles, right? Those are your lateral ventricles. Here's an MRI, right? This is, again, sort of a sagittal view. You can get a really detailed sort of image of the brain, right? You can see all those nice sulci and gyri. And here's what you might get from a DTI experiment, right? Anybody remember an image similar to this right? Now you remember. Yeah. So that was kind of exciting, right? All right, don't worry about that table. It's all information we've already talked about. Right, so that gives us structural information, right? But sometimes we want to know about activity, like what's actually going on in there, right? And what we've learned is that the brain runs on electricity, right? It's just electrical signal. So we want to think about ways that we could record those electrical signals. Maybe we could influence or manipulate those electrical signals. That might be exciting, right? So we have microelectrodes. These are very, very small, sort of thin pieces of metal that you jab into a brain. right? You can get it really close to a brain cell or even inside a brain cell. You can record from one neuron at a time. You can record from multiple neurons, right? So that's kind of exciting. Here's a rat brain with a bunch of electrodes going down in it. This is a, it's like dental cement, right? So like your dentist might use to create some, fake gums to hold your teeth. If you had dentures or whatever. And then you have these wires that go down into the brain, and it records brain activity while this rat's, I don't know, playing the cello or whatever you've trained it to do. In a couple weeks, we'll actually talk a lot about EEG activity, right? Anybody had an EEG, know what one is? They stick these things on your scalp, right? And they record the electrical activity of the brain cells that are near the surface. That changes over time. You can get some idea about what folks are doing or problems they may be having looking at their EEG activity. You could also look at metabolic activity in the brain. There are a number of ways that you can do that. Just be aware that that exists. There's also some functional imaging. Again, if we don't want to cut your head open and jab you with an electrode, right? There's a functional MRI. This kind of uses some oxygen levels to measure activity in different brain regions, right? Typically what you'll have someone do is you'll have them do two things, not at the same time. They do one and then they do the other, and you subtract the difference between those two tasks, right? So if you have them like look at some pictures, and then you also have them like next image or next task is like read, then you can say, well, these brain regions that were active during reading but were not active while they were just looking at images, then they must be the ones involved in reading, right? Don't worry about CFOS or PET scans. So what an MRI looks like, we'll talk about MRI data as we go. Sometimes you want to stimulate part of the brain. You can do that with electrical stimulus. That's pretty cool, right? So you can take one of those electrodes, and instead of like going, hey, what's happening, you can go, watch this, and like I actually pass a current through there to excite part of the brain. You can also use chemical stimulation, right? And so we talked about those excitotoxic lesions, okay, where we were exciting those brain cells to the point of death. You can stop before they die, right? And you can turn those things on or off using, for example, GABA agonists, right, to inhibit, or glutamate agonists to activate. Here's another fun magnet story, right? Transcranial magnetic stimulation. This is kind of interesting. You get a powerful magnet, you focus it on a brain region, it'll turn that brain region off briefly, and then it kind of can come back on later. So it's not, sometimes there are some long-lasting effects from this. It's actually a group in Italy who's using transcranial magnetic stimulation to help folks that are addicted to cocaine. It's kind of an interesting, yeah, right? It's kind of an interesting approach. All right, now this is kind of cool too, this thing called optogenetics. Anybody heard of this? You're using viruses again, okay, so, but you've chopped off again the part that makes you sick. You can trick brain cells into making special receptors that you can turn on with wavelengths of light. So it's just kind of cool. So you get this, you get these cells like blue light or yellow light, whatever. You can open and close ion channels. Now I know someone's going to ask this question, like, how do you get light into your brain, right? You have to make a hole right in the skull, and then you jab in a laser. That's how that works. True story. You stick a fiber optic cable down in a brain, and lasers are really powerful LEDs. You can use LEDs too to send wavelengths of light in, and then you can turn on and off ion channels, right, that you've put in there that are light sensitive. So that's pretty cool. They're making, there's another method here too. Instead of making these sensitive to light, you can put a binding site on here that's shaped specifically for whatever chemical you want. Pick a chemical not in your body normally, right? And then you can take that pill with that chemical in it. Doesn't do anything anywhere else, but when it gets around to those special receptors you tricked your brain into making, it can open and close those ion channels, right? And so that's really some specific targeted stuff. We call those bespoke receptors. I don't know why stuff keeps disappearing. It's like one of those birthday candles you keep blowing out and then it keeps lighting again. Those are irritating. I mean, who thought, you know what would be great? Cake. I'm with you. You know what else would be great on top of that cake? Melted wax. Like that's not a good idea, is it? This doesn't make any sense. You guys are thinking like, birthday candles, cakes, they're awesome, right? All right, questions about that? Methods, we talked about all that stuff. Neurochemical stuff. I mean, you can stick chemicals in the brain and stuff happens. That's really what I want you to take away. I know, right? It's like a lot of slides, but that's the end story. So I think in the interest of time and your attention, that's what we'll do. Oh, hey, these are kind of fun, though. Genetic studies, right? What about twin studies or adoption studies? Anybody heard of either of those? Yeah, these don't happen as much anymore. A number of years ago, folks thought, hey, if somebody has twins and they don't want them, right? You know, we find someone who does want them, right? That makes sense. It's perfectly fine. But the idea back then was like, well, I think it's better if we separate them and send one to two different homes, right? Send one in each direction. I know, right? Which for those individuals, it's probably not the best. For people interested in genetics and you know, how that affects behaviors and outcomes and so forth, it was really a good thing, right? Because then you have two genetically identical people in two completely different environments, and you didn't have to steal them, right? So you didn't have to run around, right? 'Cause that's another way to get this to happen, right? So twin studies and adoption studies, we sort of have what data we're going to get out of that, right? Not as much anymore. So, but if any of you do decide to have kids, try to have two at the same time, and then we can take one and, I don't know, put it somewhere else for a while, right? See what happens. You got an extra, right? Nobody thinks that's a good idea, Kyle.
Well, you told us to always have a spare tire, so we won't be having kids.
Great. I'm glad someone listened. Yeah. Thank you so much for listening to that. I appreciate it. When I get older, I don't want people clogging up the line at the grocery store when I'm trying, you know, that already irritates me now, and I know the older I get, the more it's going to irritate me, so the more people you make, it's going to be a problem. There's some other gene studies we could think about or not. Okay, questions about methods? A few things to you before we move on. Yeah, I don't see chapter 6 at all, do you? Let's just try this. There's a chapter 6. All right. Any questions about methods before we move on to vision? Great. No one asked this, but they should have, Kyle. Is chapter 5 going to be on the test? Not really, right? It doesn't mean you don't need to know things from chapter 5. I'm just not going to ask you to tell me, like, what's the difference between a PET scan and an MRI, right? I don't really care if you know that, okay? You just kind of need to have some small background there.
So our quizzes go until December. Can we take them on the ones that we get the low score the first time? Or will it just let us click on them? Because you can open them and it'll say start to begin test.
You can try it. It shouldn't work. If it does, I wouldn't tell anybody else. But now Kyle's going to check and make sure you can't do that.
Well, Kyle, I need you to do it like it takes.
Also, we dropped 3 of your lowest test scores, right? Yeah, so that happens. So some of you, like one of you, has actually done more than 3 quizzes. So I'm not going to call out that person, but that's really impressive. Now, that may have changed since I looked like a couple days ago. because I was like, hey, there's quiz grades, and then some of you don't have a grade in that current grade column. It's because we've dropped those 3 lowest quiz grades. It happens automatically, right? Once you complete that 4th quiz, then that, you'll notice, becomes your grade, and that will change as you take more quizzes, right? So there you go. So if you did like 3 quizzes and you're like, why don't I have a current grade in this class? It's because those 3 quizzes get dropped. Someone worried about that? Probably not. When are you going to think about your current grade in this class? So the final is what, the 12th of December? Probably about the 14th of December. You'll start to worry about your grade in this class. You're like, huh, I should have done something. A long time ago, like 4 weeks ago, I told you your nervous system does 3 things, right? Input, calculation, and output. Okay. Today we're going to talk about one of the major sources of input to your nervous system, and that's going to be vision, right? As a human, vision is a fairly important sensory capability that we have, right? If you don't believe me, I don't know if any of you do your own taxes. Anybody do your own taxes? There's always a checkbox there, right? To ask if you're blind. It doesn't ask if you're deaf. It doesn't ask if you have somatosensory issues, right? It asks whether or not you are legally blind. Okay, so that should tell you we think vision is a valuable and probably the most valuable sensory system that we have as humans, right? So we'll talk briefly about some of these things. Don't worry about it too much. We're going to skip a few things in this chapter. But we'll tell you what's important. All right. How many of you have ever taken a class called Sensation and Perception? It's the best class you ever took, wasn't it? No comments? It is important. How many of you have heard of sensation and perception? Yeah. Hopefully someone talked to you about that in your intro to psych class. Maybe you've just heard it while you're out on the street, right? It's a common street term. Sensation and perception. That's true. They're different, and we want to think about how they're different. Sensation is really what are those cells doing? What stimulus are they detecting, right? Perception is what do you make out of that sensation, right? You take that information and you do something with it. You make some sort of like, oh, well, yeah, I see a round thing and it's red, and it's probably an apple, right? That's perception, how you perceive that. Actually, in fact, red is a perception. If we're going to talk about vision, we're going to talk about light, right? Light, we typically think of coming in color. We think about different wavelengths. We can think about how bright or intense that is, and then we can think about the saturation of that as well, right? So if you think about red versus pink, right? So red is that dominant wavelength. Pink still has red as a dominant wavelength, but it's been sort of desaturated a little bit, right? It's been mixed with some white light. It doesn't really matter what sensory system we're talking about. There are some things that are going to be the same, right? And they're going to be things related to the stimulus and how we collect information from that stimulus and what we do with that, right? It doesn't matter if that's an auditory stimulus or a visual stimulus or a somatosensory stimulus. We're going to do the same thing, right? First, we're going to have some specialized neurons, right? And they're going to detect those physical events. So when we think about vision, we're thinking about the retina, right? We're thinking about the rods and cones. You guys heard of rods and cones, right? Think about the rods and cones that are detecting those wavelengths of light. Once we talk about the auditory system, we're going to think about inner hair cells, right? So you have these cells in your ear that detect deflections with different frequencies of sound coming into your ear. If we're thinking about your vestibular system, right, we're going to think about how stuff in your inner ear is moving and deflecting different cells, right, and to coordinate your balance and if you're spinning around or whatever. It's the same idea. You have specialized cells that are collecting this information from the outside world. Now, does your brain, once you're into your brain, does that process light signals? Does that send light from one cell to another? Does it send sound waves from one cell? It doesn't, does it? It sends electrical signals, right? So we have to go through the process of sensory transduction. Sensory transduction is simply taking whatever sensory stimulus that is and translating that into an electrical signal in your brain. It's that simple, right? Happens very, very early in the sensory process, like that first neuron, right? Okay, that first sort of interaction is going to be taking light waves and turning those into an electrical signal. Taking sound waves, turning that into an electrical signal, right? Taking mechanical pressure on your arm and turning that into an electrical signal, okay? If you notice, it says these are slow graded receptor potentials. This is different than an action potential. You guys remember that action potential, right? It was like an all or none. Okay, we're coming along, we're just up, right? We have that nice spike. We come back down, the after hyperpolarization. These receptor potentials are graded. They're slow. They kind of go up and down. You know, we're not necessarily firing an action potential. We're just moving up and down as we collect more or less of that stimulus, whatever that is, right? For this chapter, we want to think about vision. For vision, we're really focusing on the electromagnetic spectrum. Have you guys ever seen this? Yeah, Melina, the answer is yes. That's exactly all you see is the electromagnetic spectrum, right? You don't see anything else. Not unless you are like a bird or a pit viper. Then you can see, I know, right? Somebody got excited about pit vipers. Then you can see other things. So we're looking at this very narrow sliver of these electromagnetic waves from around 400 nanometers to 700 nanometers. Now there are wavelengths far beyond this on either side, right? There are things that are much larger. Those are television and radio broadcast bands. Things like radar are a little bigger. And then also infrared, right? Anybody have a device that uses infrared signals? TV remotes, perfect. Yeah, I'm really excited about that. I'm surprised you knew they ran on infrared. They used to run on sound frequencies, right? The first television remotes, I know, it emitted like high-pitched frequencies. There were some people who complained that their dogs were upset when they changed the channel. I think, you know, that, I don't know. Maybe they were really watching the news. Who knows? Infrared is also cool for pit vipers. Anybody know why they call them pit vipers? They're really great to push people into a pit with these vipers. No. They're called pit vipers because they have kind of pits like toward the back of their heads a little bit, right? And how do you know what's the head of a pit viper? It's the part that can bite you. The rest of it's the tail, right? Just in case you're curious. Because some people are going to ask, like, where's the head? It's the part that bites. The rest is tail. So these pit vipers, they have these infrared sensitive pits kind of back on their heads a little bit. What's cool about this is, how many of you have ever seen like an infrared camera? Kind of looks like a heat signature or something, right? So these pit vipers, they use heat signatures as well, like kind of like how we use vision. They have normal vision as well. It's a little different than human vision. But there's a guy who does some research on weird sensory systems in animals, and he's in Germany. What he does is he takes, it's a fun experiment you guys should try at home. He takes pit vipers and he wraps their eyes in electrical tape. Really seems like a great idea, right? So he takes electrical tape and wraps around the eyes of the pit viper, and then he carefully puts the pit viper back in its cage. And what he'll do is he'll let a rodent loose in the pit viper's cage. You know, snakes eat rodents. It's a pretty standard thing. And what's kind of cool is even though the pit viper's eyes are completely blocked by the electrical tape, they'll still accurately strike and nab the rodent. That's pretty cool, right? And you might think, well, what about like olfactory cues? Olfactory cues are great, but they're not that good at spatial sort of information, right? You're like, hey, I think I smell something over here, but I'm not entirely sure, right? This is going to be the same thing with a snake. They're not going to be quite as accurate, but they have these pits. So infrared, that's awesome. Then you get into our sort of visible part of the, it's visible to us. If you move into ultraviolet rays, anybody heard of those? Right? Yeah, those are fairly important. You should be aware of them. They exist. They don't, well, I was going to say they don't do a lot for humans. Unfortunately, they do a lot for humans, but they don't, you know, you can't see them, right? They cause a lot of damage. But ultraviolet rays, if you're a bird, and some insects actually can see ultraviolet rays, if you use special cameras to take photographs of flowers using ultraviolet light, you can see that it reveals some interesting patterns on the flowers that humans can't see, but seem to be really attractive to birds and other pollinators, right? And they're like, oh, look at that. We might just see it as a, you know, solid color flower, but there's some pattern that says like, hey, land here and stir up my pollen and, you know, help me impregnate other flowers. That's like a weird breeding system, right? Flowers. We must think about that. I'm thinking right now of like how this would work if you were, yeah, no. It's going to involve like some courier service, right? You're going to call like somebody from FedEx to come to your house and like pick up something and make a delivery to some other house, and it just doesn't work that way. See, you guys are putting all this together. X-rays, I'm not going to ask if you've had an X-ray. Again, that's no personal medical information. I assume most of you have had X-rays. That's a pretty standard thing, right? And then you get down to gamma rays. Those are great. They turn you big and green, so watch out for that. How many of you have ever seen an eye? There you go, right? Have you ever dissected an eye? Did you pull it out of the cow? It was the same. What'd you do to this cow? Just, yeah, so cow eyes, right? They're these big gelatinous clumps, right? Did you pull the lens out of there?
Yeah, the part that you can keep.
Yeah, you still have it?
I don't know.
Yeah, you have to be careful. It looks like a butterscotch candy. I'd be afraid somebody'd eat it. So your eye, what are the important parts for us? We want to think about the retina, right? So the retina is it's like a thin layer of the nervous system on the back of your eye, right? So we know a lot about the visual system. One of the reasons, Garrett, that we know so much about the visual system is it's the only part of your nervous system that you can see without cutting yourself open, right? How many of you have ever been to the eye doctor? I can tell some of you have, right? So just let's imagine you went to the eye doctor once. And what did that individual do? They actually like looked into your eye, right? And he probably had like some like magic square that he was moving around, right? And there's this like super bright light like sweeping your eye, and you're like, why is he trying to burn a hole in my face, right? That was so that individual could see your retina, right? And see the back of that. And they can actually see that nervous system tissue, right? And so it's one of the reasons we know so much about vision is we've been able to see it for a lot longer than we've been able to see the rest of your nervous system, right? So that's fun, right? Photoreceptors, you have two kinds, rods and cones. They both collect wavelengths of light. They take that wavelength of light, and through some things we're not going to even have to think about, but through some cool biochemical processes, they're going to take that wavelength, that photon, that little bit of light, and convert that into an electrical signal that they'll pass on somewhere else. There's your eye, your eye muscles, right? You have some muscles, extraocular muscles. They move your eyes, right? Everybody take a moment and practice moving your eyes. You just move them around, right? Try to cross your eyes, uncross them. Try to make one go up and down at the same time. No, I think that one's hard, right? Here's your eye. Light comes into the lens, gets reflected on the back, and focused on the back of your eye, turns into an electrical signal, goes to the rest of your brain. Some of you may be nearsighted or farsighted, right? That's a thing that happens. What happens there is your eye is the wrong size for how powerful your lens is, right? And so if you think about this projector is actually a really great example, right? If I were to take this screen and I were to move it forward, or if I were to move it back farther away from the projector, things would get fuzzy, right? Because there's this focal point where all of the wavelengths of light come together and they focus at that same spot, right? In someone who is nearsighted or farsighted, their eye is again, the wrong size for the power of their lens, and so they're either focusing too early or they're focusing too late, and then whatever hits the retina is going to be fuzzy. That's why to fix this, you put another lens in front to bend the light a little bit before it comes in so that all of the light focuses at the exact right spot. It's exciting, right? Now, some of you may wonder why you may have noticed your parents' arms are getting too short when you take them out to dinner for them to read the menu. Anybody notice this one? Right? They try to hold the menu out farther and farther. Particularly your mothers might be in that category because they don't want to get glasses, right? And they'll stretch their arms way out. As you get older, here's a real, write this down because this is going to happen. As you get older, stuff doesn't move like it used to. Okay? So things aren't as flexible. They don't bend as much. Your lens loses some of that flexibility, right? And you're not as able to move it to bend those wavelengths of light. Okay? And so as you get older, it becomes harder and harder to sort of bend things, especially that are close together, right? And so right now, if you were to move your hand close to your face, assuming you're not already having difficulty with things that are close to your face, you would, have your finger like, I don't know, somewhere here. As you get older, you would have to move your finger farther and farther out for it to be in focus, right? Because you're losing the ability to bend your lens to focus things that are closer to you. Does that make sense? So there you go. Questions about that? It's kind of a fun thing, right? What's the fovea? Anybody heard of the fovea? This is kind of a spot. They say it's in the center of the retina. It's not really in the center. It's kind of off-center, but don't worry about that too much. It's not going to make a real difference in your life. It's the part of your retina where all of your cones are jammed in there, right? And these are the things that you usually use for most of your vision, right? How many of you, when you want to look at something close, like you really want to get real detail on it, you hold it right out here to the side, right? It's exactly what you do. Like, wow, look at that. That's like the coolest thing I've ever seen, right? No. You put it right in front, right? So if you're reading, I know how many of you have read your books, right? You put it right in front of your face. Most of you are like, reading text messages from your friends about something I'm certain is important. You're going to put it right in front of your face. You don't like hold it over here to the side, right, and try to send a text message. It doesn't work that way. So your color-sensitive cones are jammed in there. It's where you're doing most of your vision. This explains, how many of you, explains this interesting phenomenon. How many of you have ever thought you saw something at night, and then when you looked at it, it disappeared? Yeah, and then you like got really freaked out and you didn't sleep all night, right? You're like, what was that? And then you look away, and then it's like, well, it's back, and you look, right? Cones are horrible at night. Okay, so Zachary, you're at night somewhere, wherever you want to be, keep it to yourself. And it's dark, okay? And you're looking around, you're using like your cones like you normally do, right? And you're like focusing stuff right here on the fovea, and you're looking around and you're like, you can't really see much because cones don't work real well. All of a sudden you see something over here out of the corner of your eye, right? You kind of look over at it. The problem is when you look over, you're taking rods, which are in the periphery of your retina. Rods are good at low levels of light, right? And you're taking and you're moving over there your cones, which are really bad at low levels of light. So you're taking something that's pretty good and you're moving over something that's not so good. And then guess what happens? It disappears. You can't see it. There's not enough light to get your cones excited. You got your rods excited, but not your cones, right? And then you look away and you're like, oh, it came back up. So it's got to be, like some night demon that's going to kill me. And so you make up some interesting story about that because you don't know how rods and cones work. But now you do. So there you go. Anybody go out at night with like telescopes and try to look at things like other people? No. Often, if you do any sort of amateur astronomy or even professional astronomy, you're gonna light is a big problem, right? Because you're trying to adjust your eyes to that. Often you will if you're trying to see something, you'll not look directly at it because it'll disappear, right? And so if you're trying to like make some adjustments on your telescope or something, it's a good idea not to look directly at it. Kind of look just over top of it a little bit. You'll be able to see a little better. So there you go. Hey, what about your optic disc? Who's heard of one of these? Due to some weird nutrient supply issues, your retina is actually in backwards. And the rods and cones actually face the back, right? So a photon of light has to come all the way through all of the cells of your retina and then get picked up by the rods and cones, and then they send their electrical signal back out, which then takes a big U-turn and comes back through in those fiber bundles, right? And that goes through your optic nerve. All of that has to be collected and punched through your fovea or your retina at some point, and that's going to be called the optic disc. This is actually responsible for something called the blind spot, right? This is not the blind spot of your car. That's a different sort of thing. But there are these tests you can get. They're typically cards. There's a small fixation point in the middle, and then there's like a circle on one side. And if you close one eye and you move that card back and forth while you're staring at that fixation point, what you will notice at a certain distance from your face, that dot will actually disappear. It'll kind of flicker out and disappear. And that's actually the location of your optic disc. You do not have any photoreceptors there at all, right? It's completely devoid of photoreceptors because we have to punch that that axon through there, all those axons, right? Those bundles of axons. Most of the time you don't notice that, right? Your eye, your visual system is kind of doing like a Photoshop fill-in kind of thing, right? Your eye is constantly moving around a little bit. We could talk about that at some point, collecting information all around that blind spot and just assuming everything that's around it is exactly what's in it, and it just kind of fills in and you don't notice it. And in fact, even if I tell you how to do this, it actually is kind of a difficult thing for some people to observe. Mostly because they don't pay attention to what they're doing. Do we want to talk about these things? We probably should. In your retina, there are a number of other cells besides rods and cones. You can look at those definitions. Basically, this is your eye. These are your rods and cones, right? Back here toward the back of the eye. This is the retinal pigment epithelium. Okay. This is a supply source for the rods and cones. These guys are doing a lot of heavy, delicate work, right? So it's kind of like they're carrying thousands of pounds of Fabergé eggs, right? So they're doing a lot of work, but yet it's delicate work at the same time, so you have to be very careful. They constantly need some replenishment from that retinal pigment epithelium. That's why they're in backwards. So that photon of light or that wave of light's got to come all the way through, get picked up, and then go through your bipolar cells. And you've got some horizontal cells here. It's got to go through to your ganglion cells. You've got some amacrine cells here. Then get into your optic nerve and on to the brain somewhere. Okay? Does that make sense? That's the test for the blind spot. It's not going to work up there. It's too far away. You could probably do this with your book, or you could print this slide out. Give it a try. All right. Here's a slightly more detailed view of retinal circuitry, right? Again, we have rods and cones. Now, this, I do want to point out, this slide and the previous one has a slight sort of lie built into this, right? You won't find rods and cones intermixed like this, right? Because all your cones are at the fovea, all the rods are around that. So the only place where you're going to find rods and cones kind of touching each other or close to each other is at the border of the fovea, right? Around the edges of the fovea and into the periphery. So other than that, this is fine. Here's your bipolar cells, these green ones, and then you have your horizontal cells here, the blue ones. They're collecting information from various rods and cones and kind of sending that information laterally, right? It's kind of interesting to know what your neighbors are doing, okay? Any of you have neighbors? Yeah, some people have neighbors, right? You guys decorate for holidays? I wouldn't either. But let's say you do, right? Maybe your parents have neighbors, they decorate, and you want to find out what your neighbors are doing because you don't want to be the person on the block who, like, you know, some people care about this, right? You don't want to be the person who doesn't decorate. So you're like, well, this person's decorating, you know, for National Cheeseburger Day or whatever holiday it is, so I should probably put out my cheeseburger flag. You guys don't celebrate for Cheeseburger Day? No. It's an important one. Information goes down. These are all those graded electrical signals. So they're just kind of signals like this, up and down, up and down, up and down, up and down, up and down. Finally, you get to this ganglion cell layer, and these guys actually fire an action potential. First cells in the visual system to fire an action potential are the ganglion cells. Up until that point, it's those graded receptor potentials, right? Not a big deal. We've already talked about transduction. Don't worry about this. Don't worry about this. I should probably tell you about something called the dark current, right? It's this evil force that runs in all of you. It says dark current. I have pretty good hearing, surprisingly. It's okay. So the dark current is interesting because the cells in the early part of your visual system, your photoreceptors, are actually, they kind of work backwards to what you would anticipate, right? So normally we think if a cell gets excited, then it's going to depolarize, right, because it's become more positive, and that's going to cause it to release neurotransmitter and then send that signal through. Photoreceptors, on the other hand, are kind of backwards. So when they get excited, they actually hyperpolarize, and they stop releasing neurotransmitter. Now, the bipolar cells, thankfully, are smart enough to know what's going on, right? And they fix the situation, and then they send the signal down normally from there. But the reason is, how many of you have ever been walking around and all of a sudden, all of your photoreceptors ran out of neurotransmitter and you stopped seeing things? It doesn't happen, right? Okay, because of this reverse situation. It could happen the other way though, right? If every time a photoreceptor was excited by a photon of light, if it had to release a neurotransmitter, think about all the photons of light like just, you know, bombarding your retina right now, Sydney. Think about that just for a moment. Okay, now, every time that happens, you've got to release a neurotransmitter. Eventually, you're going to get fatigued, right? And so, is it okay if your photoreceptors get fatigued while your eyes are closed? Well, who really cares, right? Your eyes are closed, you're not seeing anything anyway. So if they run out of neurotransmitter while your eyes are closed, it's not a big deal, right? But if they run out of neurotransmitter while you're kind of crossing 20th Street, that probably becomes a big deal, right? Because I'm probably going to be driving along and I'm going to yell at you for not watching where you were going. Okay? I always joke if I ever get hit crossing 20th Street, I'm going to donate my life insurance money to build a pedway. And just name it after me. Just make sure that happens. Okay, we should talk about receptive fields briefly. Here's what I want you to get out of this. Every photoreceptor is responsible for a small bit of the world. That small bit of the world that it's responsible for, that's its receptive field. That's it. Okay. Anybody who's a baseball fan? Anybody play baseball, softball? Ever seen those sports? Know they exist? Okay. Yeah. So you've got a field. It kind of looks like an ice cream cone. That's one reason I like the sport. You never thought about that, did you? Looks like, and so down here's home plate, and then you've got your, your other bases, and then you've got a guy out here. We're going to call him center field. He's kind of responsible for a certain area, right? That's his receptive field, right? That's the area of the field where he's got to catch the ball if it comes in there, right? And then let me make sure I've got this. Over here you've got your left fielder, right? And over here you got your right fielder. And these guys are responsible for other parts of the field, right? And so you can just call this guy a photoreceptor. And it's responsible for photons that fall on that part of the retina, which corresponds to that part of the world, right? That's a receptive field. Now, who remembers that story I told you about the fovea and like putting important things on the fovea, right? So if you're putting important things on the fovea, you want to have receptive fields. You want to have what we call acuity, right? That is as detailed as possible. So we're going to structure things differently, right? So one photoreceptor is going to contact one ganglion cell. So if this ganglion cell fires an action potential, what do you know, Maddie? You know there was a photon of light right there, right? That's exactly what you know. If this ganglion cell fires an action potential, what do you know? Well, one of these like 6 or 8 photoreceptors caught a photon. So it's a little fuzzy, right? Okay, it's not quite as detailed. So this is going to be in the fovea, and this is going to be out in the periphery. All right, so that kind of gets us out of the eye. There's not a whole lot of there's a lot of fun stuff happening there, but there's not a lot of the sort of higher order things, right? There's a ton of your brain that's devoted to processing visual signals. One of these areas is called the lateral geniculate nucleus. Like 2 weeks ago, I said, hey, we're going to talk about something called the thalamus. Wesley, I believe you asked me, what does the thalamus do? And I said it was a sensory relay area. It does some sensory processing. I'm not sure it was you, to be honest, but let's imagine it was. The LGN is located in the thalamus. Why is it called the LGN? Well, it's lateral. It's also actually called the dorsal lateral geniculate nucleus, so kind of on the top, on the lateral side. Geniculate, genu is means knee, so it's got this kind of bent structure in humans, right? So it's kind of this bent. See how that kind of looks like a knee? Just imagine. So information comes into the eye. Some crosses over and some doesn't. I know everybody's like, oh, left eye goes to the right side and the right eye goes to the left side. It's not exactly true, right? It's really what's in the right visual field goes to the left side, and what's in the left visual field goes to the right side. I know that sounds exactly like what I just said that wasn't true, but you can fix this by like closing one eye briefly, and you can realize like you can see some stuff on the other side of your nose, right? Okay, so that's like the other visual field that's not going to get crossed over. There's a whole thing I could tell you about nasal and temporal retina, but you don't have to worry about that. Just close one eye and figure out where it goes. Don't worry about this slide. And that's the same slide. All right, LGN has 6 layers, very similar to the cortex. It's actually got more than 6 layers, but we decided not to fix that after we found out there were more than 6 layers, so we're just going to leave it at 6 and just kind of put things in the middle, right? You've got something called the magnocellular layers. They process things that are larger receptive fields. You have the parvocellular layers. Those are smaller receptive fields. And then you have the coniocellular layers. These are, well, that's from the Greek word for dust, right? There are these layers in between that look kind of dusty. When you look at the LGN, they actually might be really important for processing color vision. See, there you go. That looks exactly like what I just drew and told you looked like a knee, doesn't it? You thought I was bad at drawing. It's just how it looks. Hey, here are a couple fun guys. These 2 guys won a Nobel Prize, David Hubel and Torsten Wiesel. Okay? They won a Nobel Prize because they figured out something really interesting. They figured out how a very small set of receptive fields that are like this big, really, okay, can all be put together so that you can go, hey, that's a door, and not just a bunch of little dots, right? When you look out into the world, do you see just all kinds of small little dots? No, you don't, right? You see integrated contours, and you're like, hey, that's a doorknob, by the way. And I'm going to put the hinges here just so you know it's a door. Put a little window on it. Okay, so you know, hey, that's a door to a house. Okay. so you know what it is. You can integrate these contours, right? And these two guys figured it out. It was really pretty impressive. They figured it out by taking a cat and like jabbing an electrode into the cat brain. And like, hey, look at this cat and see what happens. And it's really fascinating. Does Ilson still make you guys watch the video of their experiment? Have you seen it? This is a life-changing video. You guys want to watch it? Yeah. I don't see the relationship there, but this is a fascinating study. All right, let's see. All right, so we will watch this video. I don't want you to take a whole lot out of this, except that what these guys are going to do is they are going to... oh, I don't want to skip there. You're never going to see the cat, so I hope that makes... I know, right? Some people are sad about that for some reason. So this cat has... remember those electrodes we talked about in that sort of method section, right? This cat has an electrode in its visual cortex, okay? Kind of the outer part of its brain. And these guys are going to show it a different visual stimuli. They're going to find its receptive field. So they're going to find the location out in space for which this one cell in the back of this cat's brain is responsible, okay? And they're going to map out the properties. What they're using here, Jess, is that same thing that guy used to like blast your eye when you went to the eye doctor. They're using an ophthalmoscope. They're going to move it around. And what's really fascinating is they're going to take those electrical signals that are coming out of this cell and they're going to send it into a speaker, and you're actually going to hear the action potentials of this particular cell as it gets excited by this stimulus. So this is pretty exciting. Okay. Those are just sort of random, just listen. This. So I told you that receptive fields were typically circular, right? But that's in the retina and the LGN. By the time you get back to cortex, you're taking all of these little circles that look like donuts, and what's better than a donut is a box of donuts, right? So we're putting all these donuts, I know, right, to make a box. I mean, if somebody gives you the option donut or box of donuts, which are you taking? Taking the box, right? It's easy. Even if you don't want to eat them all, you can just resell them. There's some profit to be made. Now this particular cell is getting excited by a bar of light oriented at a certain degree, right? So that's pretty awesome. You have cells that are excited by different orientations, which is how you can tell that there's like two upright sides of a door, right? So this is all you have to do to get a Nobel Prize. Actually, they had decades of work on this, right? Like really trying to get answers, and no one else was doing it. So, like, if you were to replicate this, you're not going to get a Nobel Prize. There are some inhibitory regions. That's what these triangles represent. We could talk about that in some detail if you guys would like. Yeah, it was. If you want to stand up and applaud, that's fine. I mean, one of them will appreciate it. The other one's dead. Yeah, unfortunately, David Hubel died a few years ago. But they did a ton of work looking at responses from visual cortex. The real point I want you to take away from that is, like, here's a receptive field, right? Like in the retina, and there's like excitatory regions and then inhibitory regions. What your cortex is doing is taking a bunch of these, right, and taking that and putting it into excitatory and inhibitory receptive fields. See how that works? It's pretty exciting. Don't worry about blobs, right? Cytochrome oxidase. It's an interesting story, but not something I'm going to ask you about. All right. So we started in the retina, little dots of light. We made some donuts. We took those donuts, we put them into a box, right? So now we have like, hey, I can tell if there's a bar of light. What does a bar of light do for you, right? Like not a whole lot, right? We need to take those bars of light and we need to put those shapes together. We need to make them into something important, right? We have higher order brain regions that do that. That's called extrastriate cortex, okay? If something is striate cortex, striate means striped. If it's extrastriate, that means it's got even more stripes, right? No, it just means it's beyond the striate cortex, right? It's the other parts of the cortex on up. And so what we call primary visual cortex or striate cortex or V1 is kind of that, you know, bars of light business, right, that we just talked about. Kind of looks like a butterfly. When you go beyond that, very, very creative with their names. They call them areas V2, because that's the second visual area. Then there's V3, that's the third visual area. There's V4, anybody know? That's the fourth visual area. I know, right? It's really, really getting exciting. It is arranged in a hierarchical way. We have this thing called a dorsal stream and a ventral stream. I'm not going to get you too much on that. People will sometimes. You know, you're processing different kinds of information in different directions. There are two broad questions when it comes to vision. What is it and where is it? Both of those are important, right? How many of you have been to a zoo? Yeah, and you saw a lion, and that was cool. And you knew what it was. It was also important where it was, like on the other side of that fence and the moat and the bars, right? That's important. If you were, you know, walking down the street and there was a lion, you can still identify what. Whoa, that's a lion. That's cool. Not quite the same kind of response, right? Because where it is different. You have to think about location. Don't worry about this. This is basically the same stuff I was just showing you with that awesome video. All right. You don't have to answer this, but anybody have a color vision deficit? I'm not anticipating a lot of hands to go up. No? All right, great. As a human, you have what's called trichromatic vision. So you have three types of cones, right? Ones that are sensitive to short, medium, and long wavelengths of light. Often we think about those as like red, blue, and green. You guys have heard about that, right? The colors are more of a perception. The wavelengths of light are more accurate. Caitlin, you can tell them all about that at some point after class, right? Yeah. So there are different kinds of trichromatic problems. The most common is what we would call like red-green, right? Typically this is seen in males. The reason it's seen more often in males than females is it's linked to the X chromosome, right? So how many of you have an X chromosome? That's everybody, right? There's important stuff on that chromosome. If you don't have it, you don't exist, okay? How many of you have two X chromosomes, right? I can figure that out pretty quickly just by taking a quick peek around, right? You can figure out who's got two and who only has one. The nice thing about having two X chromosomes, if one of them's screwed up, just use the other one, okay? If you only have one X chromosome, you have to use that one, right? And unfortunately, that's where color vision is located. And if you've got a screwed up X chromosome, that's the only one you got because the other one you got was the Y, then guess what? You're going to have a color vision deficit. If you get 2 X chromosomes and one's kind of screwed up for color vision, just use the other one. It's fine. Okay? Does that make sense? So that's why we call it a sex-linked issue, or sex-linked genetic trait. Red-green is the most common. Red and green photoreceptors are very similar structurally, right? And if the structure of one is tweaked just a little bit, it brings that peak sensitivity closer to the other, and so the ability to discern red from green becomes more difficult, right? Because you start to, their sensitivities become more similar. Blue is a completely different thing way off on the side. Questions about that? I went to high school with a girl who was colorblind, and I didn't realize how awesome that was. I thought she was just like the weird girl who wore brown pants all the time. Because she did wear brown pants all the time. She said she always bought brown pants and shirts that go with brown pants because it would always match. Because she didn't have to ever try to match another color. She didn't know what color things were. She had red-green color blindness. Didn't realize how genetically weird that was. Missed out a real opportunity there. Just to talk to her about color blindness, nothing else.
Neon colors well, or blues. So if he'll buy stuff like prom in high school, he'll buy stuff to go with my prom dress. And I'm like, these aren't the same.
Yeah.
And he only wears brown pants as well.
There you go, right? It's a safe choice. So now you're going to be looking around for people who wear brown pants all the time. You're going to notice, like, brown pants today. Keep a journal. The problem is they may think they're brown all the time, right, even if they're not. Right, Kyle? Yeah. This is a real problem in sports. I remember when I was in high school, my high school team was green. One of our big, like, close rivals was red. And there was a guy on our basketball team, he was a little older than I was. He was red-green colorblind, would sometimes pass the ball to the wrong people because it looked like they were wearing the same jerseys. So there you go. You should find out next time you play tennis, like if your opponents are red-green colorblind and then like have like red strings so they can't see the ball coming off. Do you think about that stuff? I think about it all the time. There you go, right? It'll give you a competitive advantage against like the 3% of people who are colorblind. I don't know that it's going to be worth it. There's some guy, his name was Young, he was right, don't worry about it. Here's what I was talking about, those peak sensitivities. Okay, here's red and green, right? And see how they're very close. If you shift one of these peaks closer together, you can see where there's that overlap. Blue's way out here by itself. You really got to screw up blue to get it over there and get it confused, right? All right. Here's a quick color blindness test. Those are the numbers you should see, right? I don't think that's exactly right. You should hopefully see 29, 5, and 1, right? If anyone has trouble, it's probably going to be up here. Again, that's more for that red-green business. Even for folks who are not colorblind, the 1 is probably easier to pick out, right? Simply because it's, again, those peaks are farther apart. Anybody just figure out they're colorblind? That's never happened. I always like hope for that moment because I don't know if they're going to be so excited because like that's pretty cool, or if they're going to be disappointed because they had other plans in life. All right, so there's some business about yellow that I don't want you to worry about, but it's in your book. If you're really interested about this, what should they do, Laura? They should send an email to you to sign up for this study. But what else should they do besides that? Yeah. Read it, or for those of you really interested, we actually have a a sensation and perception class, right? PSY 324. They're going to offer it in the spring. So for anybody who's going to be here in the spring, I've heard the guy who teaches that's pretty good. So you should check it out. I teach that, Kyle. Okay. I knew you did, but I just want to make sure they did. So nobody was confused. Hey, how many of you love after images? Kind of fun. Everybody seen one of these before? So what you can do is you can stare at that apple, I don't know, probably like 40 minutes or so, and then look over at the blank spot, right? I'll give you plenty of time. Go ahead. Hopefully what happens in maybe just half a minute or so, right, of looking at the apple and then moving over, what you see instead of a green apple with a reddish pink sort of leaf, you actually start to see a green apple, no, a red apple, I'm sorry, with a green leaf, right? Did anybody see that? So how many of you have ever been tired? Yeah, it happens, right? How many of you have ever competed in a sports competition and didn't get kicked out for scheduling difficulties? Yeah, so what sports competition was that? Yeah. Basketball? Yeah, so let's say you're playing basketball, right? And let's say your normal level is like here's a scale of your basketball skill, right? I don't know where it is. We're just going to put you up here, right? So like here is like, you know, your great-grandmother. Although I should tell you, my great-grandmother actually played basketball when she was in high school. She wore like a skirt and leather. It was really weird. And up here, who's probably the greatest basketball player ever? I'm really excited about this, by the way. We got to put Clyde Frazier up there, right? No, nobody's excited about. He's got some new shoes coming out from Puma, and I'm just really kind of. Nobody's excited about those. No, nobody knows Clyde Frazier. That's fine. He's also talk about a guy who can't match his colors. Huh? It's very unique, right? Yeah, it's a very unique style. Still, he's. I don't even know where most of that stuff, you know, it's custom made. Like he goes and picks out the fabrics and then has someone make the suits for him. Like every year he spends like so much time like walking up and down the streets in New York, like going into fabric shops, buying like big bolts of fabric so he can get those ties and suits. They're like custom only for Clyde Frazier. So, but we're going to put you here, which that's not so bad, right? I mean, that's pretty far up the list. But let's imagine one day you're a little tired. And instead of playing up here, you've moved down to this level, and then some guy who's not that great comes along and he beats you because he looks better because you're tired, right? And that's actually what's happening here. So when you stare at this image for a long period of time, all of the green photoreceptors that are in that space with that apple, they get tired. And when they get tired, because they're matching up against the red photoreceptors, when you look away, they look like they're actually being activated because you're comparing red and green, and you're like, well, whichever one is more active is the one I'm seeing, right? And if the green is just really, really tired, Ebony, and it's down here, it's not doing anything, right? The red is going to look like it's really powerful and coming through, even though you're just looking at a blank white square. Does that make sense? Same thing works with motion after effect, right? It's the same idea. If you watch something move in one direction for a long period of time and then you look at something else that's stationary, it'll look like it's moving in the other direction, again, because you're comparing that movement, and then one set of cells got tired, and the other set, even though they're just kind of accidentally active, it'll look like things are shifting in the other direction. Don't worry so much about this. Don't worry about that. Okay. Let's talk about studies with humans, right? And let's talk about that extra striate cortex, what these brain regions do, what happens when they're damaged, how we figured out these things happen. There's a study a number of years ago looking at folks that they'd show them images. Some of those were what we call achromatic, right? So think of like a grayscale image. And some of them had color information. There's a brain region called V8 that consistently was excited only for those color images, not for the grayscale images. Additionally, there were 92 folks who are unable to see color. Now, it's important that you look at their retinas to make sure their retina is intact, right? Because you could have a retinal issue and it's not an extra striate cortex area. And it actually amazingly matched up perfectly in those 92 folks. They all had damage to their area called V8. Now, how do you get damage to area V8? Typically, we're probably thinking about things like what we call a cerebrovascular accident, right? Nobody has one of those on purpose. We're talking about some sort of aneurysm or stroke situation, right? Can cause some brain damage. Based on the location of that, you know, where it's sort of, let's go back up. Based on the location of this, it might be difficult to have a head injury, but head injuries can cause this as well. All right. Don't worry about this one. V2 is all about form. Spatial frequency, I'm not going to ask you about this, but it's kind of interesting. There's Abraham Lincoln, in case you didn't know. Anybody heard of him? He's sort of important. Right now I want to make a joke about how he's the most reptilian president of all. I think like 2 people in this room, and I'm both of them, are going to get that joke. There's a brilliant television program, Laura, you should watch called People of Earth. It's about a support group for people who've been abducted by aliens. There's one guy who thinks all these presidents are like reptilian aliens. And he's asking him about, like, you know, notorious presidents like Nixon, for example. And then he asks him about Abraham Lincoln. He's like, oh, yeah, that's the most reptilian of all. This is a really funny, dry line, and you should watch it. It was on TBS for like a season and a half. It didn't make it any farther because I was the only one watching it. Is it? Maybe that's where they got it from. I don't know. That's a good show, though. Thank you. I'm going to give you a Planet Express. That's exciting, right? Let's not worry too much about form. That, by the way, was a monkey brain, not a human brain. We should talk about a few things here, though. How many of you think faces are awesome? They are. That's how we recognize each other, right? Humans actually have a brain region where we're sort of gets really excited when we see faces. So that's pretty cool, right? So you see someone's face and you're like, whoa, there's a face. Now, if you think about faces for a moment, they're really all the same, right? They all have eyes and noses and ears, and there's not much difference, right? If you're like, draw a face. There's a face, right? I'm done, okay? And it doesn't matter who you are, you look at that and you're like, that's a face, right? And if I can do that and you know it's a face, then they're all the same, right? But some people's faces are a little different. Some people are faces like this. Right, and then some people have faces like this. I don't know. That person looks a little nervous. There's a lot of individual variation in faces, right? This is how we recognize people. We don't go like looking at elbows and go like, let me see your elbow. That's how I know who it is. We look at faces, right? And so we actually have a brain region. It's called the fusiform face area because fusiform just means weird shape, and you know, face, it detects faces. It's pretty obvious there. What's really interesting about this, though, is that it probably is not specific for faces, right? It's probably specific for anything that's like fine differences in something, right? So musicians, for example, or instrument collectors, right? Even if you're not a musician, if you show them different sorts of instruments, right? Like maybe like different violins, and we all look at that and we're like, well, that's a violin, I guess, right? Whatever. They might be able to tell you different things about the model of that violin, when it was made, based on like the curve of the neck or the shape of the body or the cutouts or something, right? So if you think about if anybody is like a collector of something, or if you're really into a particular sort of thing, cars, for example, right? That might be something like some people can tell minor differences in headlight assemblies or something from one car to another, and they can tell you some information about that. Owls, anybody into owls, birds? Oh, ornithologists. So the fusiform face area will get excited about that, right? I mean, if you see a bird, you're like, well, that's a pigeon and that's not a pigeon. Like, that's how I know birds, right? But somebody who's really into it, they can tell you all the different species of pigeons or something. Nobody's excited about pigeons? No. Looks like what? Yeah, there's like this little... He's got to have hands, right? Is that better? He had a little bit of hair, didn't he? I think so too. No. We also have brain regions for looking at body parts. So this is kind of cool. Fusiform face area gets excited by looking at faces. It also gets excited by headless bodies. Now, I know, right? Everybody's like, oh, headless bodies. Now we're talking about something. I would ask how many of you have seen a headless body. You could raise your hand for that. Was it the cow?
No, it was in Morgan.
Oh, okay. That's okay then. So your fusiform face area got a little bit excited about that. And body parts, right? Like random body parts. But you also have a brain region called the extrastriate body area. Gets even more excited for those headless bodies and body parts, right? So if you just like randomly see a leg somewhere, that brain region is going to get excited about it. It happens, right? What if somebody's hiding behind a tree and they just like stick their leg out, right? Like that could work. Doesn't have to be like detached, I suppose. Don't worry about the hippocampal place area. We should think more about faces, though. How many of you have seen a lot of faces? Like everybody, right? How many of you, when you were like 7, had seen fewer faces than you've seen now? Yeah, that makes sense, right? So what's really interesting is if you look at the development of the fusiform face area over time, here's an 8-year-old child, and here's an adult. The fusiform face area actually gets bigger, right, over time. That tells you it's an experience-related change, right? Okay. Yeah, it's already there. It's already sort of pre-excited about faces, but the more faces you see, the more excited it gets and the better you get at differentiating between two people, right? You can always use a faulty fusiform face area to get you out of trouble, right? You should try this. Like, you know, I really thought that was you. My fusiform face area doesn't work very well. I got a little confused. So if you use that successfully, let me know. I'm always interested to try that. Retinal disparity. Hey, most people have 2 eyes. Those 2 eyes are separated by some space in the middle, right? Like the width of your nose, the bridge of your nose, right? So they're like separated. Because they're separated, they give you 2 slightly different images of the world, right? And what you do with those 2 images is you sort of blend them together to get some 3-dimensional information, which is kind of interesting. A lot of depth perception actually just works with 1 eye. So, you know, if you want to try this, walk around with like 1 eye closed, and you can get around fairly well that way. Not perfectly, but you can do it. Hey, who remembers these guys? Yeah, I showed you their names twice, right? That probably means something. I'll probably ask you about them, right? Because they're kind of cool. Orientation selectivity, don't worry about that. We talked about it. What about optic flow? I really thought I shut that door. How many of you have ever moved? I mean, not like your house, but just like through space, right? Like everybody. That's how you got here, right? Because I've been by this room and it's been empty before, so I'm going to assume none of you were here at that time, right? So you had to move to get here. So visual motion is probably one of the most important types of motion that you can have. Most of us can imagine a world where we're not seeing color, but can you imagine a world where you don't see motion? That seems pretty weird, right? And there are folks, though, who don't see motion, right? And so it's kind of weird, right, Cody? Like, how do you think about, like, not seeing motion, right? It's like all you think about is stuff that moves in your visual field, whether it's moving itself or you're moving in relation to that, right? And that's that optic flow business kind of moving past you. But there are folks who have the inability to perceive movement. There's a brain region called MST. There's another area called MT. Both of these are involved in visual motion. Okay. If you have damage to them, you're not going to be able to perceive movement. That's going to be a real problem. Okay. It's going to be a real problem, right? So you can imagine, like, if something moves, it's gone. You don't see that. You don't see that it moves at all. Only things that are static. Now the problem is things are only static if you're also static, right? Because every time you move your head, now stuff's moving. Oh man, it all disappeared, right? So you can try to think about that. It becomes a very, very difficult situation. All right, and there's where that's located. So don't worry about that too much. And don't worry about this. And that's just the summary. All right.